And then with technology, it is the application of the knowledge that was gained. And then human development is the process of enlarging people's freedoms and opportunities and improving their well-being. Now, the way that the United Nations has structured this is by using the Sustainable Development Goals in developing humans. So the Sustainable Development Goals are broken up into 17, and I will try and touch on a few of them in my talk, including no poverty, no hunger. I will look at renewable energy solutions, as well as climate change and well-being. Now, I think we both, or rather we are all aware about climate change that has been occurring and how it has affected the agricultural sector, and by extension, the normal people on the ground, which is you and I. This means that the amount of food that is produced has decreased because of the effect of the climate change, which has caused the soils to become more saline or alkaline, also causing there to be drought. In our hotel, there was a notice that was placed there to be mindful of how we use water. And in Cape Town, there was a day that we called Day Zero, where we were warned that there was going to be scarce water, which meant we had to be mindful of every drop that was being used. And so plants have a way of adapting to these stresses, which we call abiotic stresses, such as drought and salinity. And the way they do this is by developing reactive oxygen species, also known as ROS. And as ROS accumulates, it causes nucleic acid damage, increased lipid peroxidation, protein degradation as well. And this the plant does in an attempt to weather the storms that it is faced. But as you can appreciate, most plants do not actually survive abiotic stresses, although there are those plants that do have tolerance mechanisms against abiotic stresses. And they do this by producing osmolytes, such as proline, which actually scavenge reactive oxygen species, causing the plant to have a bit of water in order to survive. Because when they are faced with drought, you will notice that the plant usually tilt and look like they are dragging themselves in order to survive. And then one of the other things that they produce in order to be tolerant to the abiotic stress are ROS scavenging enzymes, which include Apex or ascorbic peroxidase. And one such plant that is able to withstand abiotic stresses, such as your salinity as well as drought, is known as sorghum. If you grew up in South Africa, you will know sorghum. We use it to make umqomboti, or also to make hot porridge in the morning. And one of the uses of sorghum as well is as a biofuel because of its high sugar content in its stem. Now, in my study that we were busy with that was concerned with sugar-grazed sorghum, which is a sorghum type that is known to have a high sugar content, we were interested in finding out whether sugar-grazed sorghum, like other sorghum, can withstand the abiotic stresses. And we published this work, and if some of you are interested, you can look it up. And the conclusions that we saw in our study was that there was reduced biomass in the sorghum because of the abiotic stress that we had exposed to, as well as the chlorophyll content. Now, this happened because when the plant is under those abiotic stresses, it can no longer take up the carbon dioxide to use it to convert it to chlorophyll that can be used by the plant in order for the plant to survive, which is why it discolors, as well as the relative water content gets reduced. Then there was increased hydrogen peroxide, lipid peroxidation, as well as increased cell death. Now, the end goal of our study was to actually have to bioengineer plants that would be salinity as well as stress tolerant. But the question that I want to pose to us is, why are we most of the time running towards bioengineering plants instead of looking at plants like sorghum, which can already withstand these harsh conditions? Going on further, I would just like to introduce sorghum to you as one of the alternatives, because when I spoke earlier, I told you one of the sustainable development goals that are important is health and well-being. And if you notice, even during lunch, one of the things that we were given was Coke. Coke has a high sugar content, which is known to increase inflammation in our bodies. And as that happens, with the increase of inflammation, there is also a direct increase in diseases. And also, it is also known that it affects the gut microbiome. So when the gut microbiome changes, that also introduces certain diseases that most of us are exposed to right now, including eczema and cancers, as well as cardiovascular diseases. So sorghum is one of those plants that can relieve inflammation. And I think most of us can know that when we go to the doctor, the first intervention that we are given is pills. And most of those pills have side effects which are associated with them. But if we use food as our medicine, we will not have those indications that the medications have. So if you are hypertensive, right now you can use sorghum as your diuretic and consult your doctor, of course, to check that your hypertension has gone lower. And I would like to also look at the impact and the benefits of sorghum. One of the things that has happened over the years is that people have strayed from the plants that are actually indigenous to Africa and have imported other plants into Africa. And this has also affected the ecosystem, as well as impacted on accessibility of food, which is a serious problem because we know that in South Africa itself, there are people who are staying in slums as we speak, and they do not have access to food, which is a basic human right. And as I mentioned before, that there is, because of the climatic changes, there is less water that is available. So if we use plants that are known to have inherently these properties, then we will not need to introduce more water into the system, and people's health will be improved. In terms of the future directives and calls to action, I would like for us to think about African solutions for African problems, because more often than not, we look to the West for our solutions when we are the ones who are having the problems. There are people who usually say that the person who feels the most of pain, if your shoe is squashing your feet, you are the one who is feeling the pain the most. So you cannot look for a solution when you are the one who is affected. Collaboration. There is a tendency for us as researchers to stick to what we know and not involve other people. As an example of what we were studying here, which is plants that can withstand drought and salinity and bioengineering, more often than not, we do not look at how these are going to impact the ecosystem at large. And then in terms of funding, we are always waiting for funders from outside instead of spearheading our own funding. And with that, I would like to thank you for your attention.
When you see me standing, I won't stop you. Ne? It's just a way of telling you that, yeah, you must wrap up. Ne? And I think she did fantastic for the time. I'm going to open for questions. She needs to get some questions if she has to get an award today. Any questions? Thank you.
All right, thank you for that fantastic presentation. My question is, from the title, I think you are likely to use omic analysis. Am I right? Yes. Okay, I just want to know which of the omics, is it metagenomic or proteomic, and why do you choose to use proteomic among the omic analysis?
One of the things that happens during abiotic stress is that the proteins themselves are actually converted. And one of the things that we know is that the transcriptome does not actually translate to the protein that you are going to find in the end process. So we looked at the proteomics to know the type of proteome that is going to be available once the plant has actually withstood the stress. Yeah.
Another question still? Don't be shy. You need to have to ask questions. I try to make sure that I give you guys a chance to ask questions. I'm just facilitating your session. I don't want to dominate it, so I want you guys to ask questions. Anyone with a question? One more question?
Thank you for that nice presentation. Please, I want to ask concerning GMOs. I believe some of this work you guys are doing is one way or the other influencing the GMO. So are you not thinking there's going to be a kind of future problem as a result of all this changing in the genetic formation of plants? Thank you.
So one of the things that I actually wanted to touch on, but because of time couldn't speak to, was the issue of integrity in science. So there are studies that are increasingly showing that the genetic modification of things, whether it's plants or animals, actually has an effect on the people that are consuming it. So even with the increase in diseases that we are seeing at the moment, some of them can be associated with the increase of genetically modified food that we are consuming. And unfortunately, there is a lack of accountability right now, and a lot of censoring of people who bring these things out. So it is one of the things I think we as people who are coming up need to address in terms of having people to be accountable and actually going back into the science to look at the impact of the things that we have produced. Because it is not enough to say that A, B, C, and D can withstand abiotic stress, but not look at the downstream effects of what we have produced is doing. Thank you.
Thank you very much. We can clap hands for her. Right on time. Thank you very much. I'm trying to be strict with time, exactly 15 minutes as it is, so I'll try to be strict. My next speaker, I think, is Londani. Okay. So I'm going to try to be fair as their gifts to do. I'll be strict, exactly 15 minutes, so that there is fairness. 2 questions at most will be. If you finish 8 minutes, 1 more question, you finish by 8 minutes, and I end it.
Time, I don't know. Go to the next slide. Oh.
Start. Are you ready?
Not yet. Anyway, good afternoon, everyone. My name is Mketo Ndani. So I'll be presenting to you a topic called tobacco use and behavior among South African adolescents and young adults. So these findings are from a systematic review and meta-analysis. So without wasting time, so I'll give you a brief background of why tobacco use. So tobacco use, as you may know, is one of the most significant public health concerns for both users and society. So also, as you may know, tobacco use has been recognized as one of important risk factor for non-communicable communicable diseases and premature death. So globally, so that is not globally. So, but even though that is the case, most of people, or there are more than 1 billion people who smoke daily, and 80% of those people, they are found in low and middle income countries. So it is also known that majority of people who are adult smokers, they start smoking at a very young age, 18 years, and they continue smoking through their lifetime. So yeah, I'd like to bring it to South Africa. So in South Africa also, it has been checked and known that tobacco, there's been a higher prevalence of tobacco smoking among all ethnic groups since 1976, and it has been high throughout the continent. So according to one of the surveys which was conducted in 2021, so it was found that 12.7 million people aged 15 and above were currently they were current smokers, and it made up to 1/3 of the adult population. And also, in 2017, tobacco contributed to 20% of total death, and if we were to quantify that, each and every year it contributes to almost or more than 80,000 deaths. And it's through the different type of disease, including TB or tuberculosis and other type of diseases. So if the use is not controlled, this number may rise to around or more than 250,000 in future. So that's why studies like this are very necessary, so that we can eradicate or try to mitigate the tobacco use, especially among young people. So these studies also can help in terms of better tailor to create better tailored intervention, which will specifically focus on young people. So the aim of this study is to conduct a comprehensive review of published literature about tobacco use and behavior among the group, which is adolescents and young adults, and to determine aggregated or pooled prevalence, so that at least you can use different type of studies to calculate the prevalence. So the method, this study is a systematic review and meta-analysis, and also it used PRISMA guidelines, and also the data source or databases which were searched are listed there, including Google Scholar, and we searched from the year 2000 till last year, 2023. And the reason for searching from year 2000 is because There's been, South African government passed the law, passed the tobacco control legislation in 1993, and it was amended in 1999. So as part of the law, it was to ban tobacco advertisement and also sponsorship, and also to restrict smoking in enclosed public places, and also to prohibit young people or minors to buy alcohol, to buy tobacco, sorry. So also, the eligibility criteria for this study, we're looking at cross-sectional studies. Also we were looking at the young adults who are aged 12 years and 20 to 24. So as part of methodology also, some studies were excluded from this study. So studies which were conducted before year 2000, and also studies which looked at electronic cigarette, because it's not always tobacco, unless if they're smoking the electronic cigarette, which includes tobacco, but we won't know. So it was excluded also, and also if it was not a full articles, let's say maybe just an abstract, it was excluded, and also if it was published as books or conference proceedings and not being peer reviewed, and also if it was qualitative or lack quantitative details, and also if it was designed as longitudinal or randomized clinical trials or systematic reviews. So two independent reviewers were they scanned the downloaded articles, and also the bias, the risk of bias was analyzed using risk of bias as assessment tool for non-randomized studies, and also to check the quality of methodology, we used Joanna Bridge Institute checklist, and also we use heterogeneity of prevalence estimates. So sensitivity analysis was also used, and final plot was used, and meta regression analysis was used. So I'll talk about that as I present my results. So what we are looking at now is the PRISMA systematic review. So 73 articles were downloaded, were searched, and there were no articles from other sources. And also after removing the duplicates, then we were left with 57. Then after scanning further, we were left with 45. Then after removing other articles because of specific reasons, we were left with 12 articles. So these are the articles which were included in this study. So studies included in the meta-analysis were conducted between 2003 to 2019, and 8 studies were conducted among secondary school learners who are doing grade 8 to grade 11, and 2 studies were among university students, and 1 study was school dropouts, respectively. So the age range for all the studies range from 14.6 years to 21 years. So all the selected studies reported current tobacco use and were cross-sectional. 7 studies used probability sampling, while 5 used non-probability sampling. And 3 studies were conducted nationally, and 3 in Western Cape, 2 in Limpopo, 2 in 3 provinces, which is KwaZulu-Natal, Western Cape, Mpumalanga, and Gauteng, and 1 in KwaZulu-Natal, and one in Gauteng. So there was no study from Northern Cape, Northwest. So Yeah, so this is now the final plot for both gender. So 22% was the aggregated prevalence, or it was the pooled prevalence. So for male, we had 26%, and then for female, it was 13%. Then, as part of the results also, the highest prevalence of current use was reported in studies among school dropouts, and the lowest was from secondary school learners. And then sensitivity analysis also was used to show that there's no evidence of publication bias according to the visual inspection. Also, the meta regression analysis was used, and also both moderators, which were sample size and year of study, showed that no impact to the prevalence, to the total, or to the pooled prevalence. So also the subgroup analysis shows that there was an increase for both males and females between year 2000 and 2020. And there are some limitations that I'll just scan quickly. A few quantitative studies countrywide. There were few quantitative studies countrywide and no studies in some provinces. And also, this study only searched published studies and may have missed important tobacco use evidence from grey literature. Then this is my conclusion also. It is very important. It is a very important study when you look at the policy making, because obviously it can help in terms of creating policies which can help in eradicating or mitigating tobacco use. So I won't go through my last slide. I'll just share the last point there to say these findings offer valuable insight for the evaluation of further policy development and may serve as useful reference for other nations dealing with similar public health concerns. So these are my reference, and the last point I would like to say is to say technology-based interventions to eradicate tobacco use among young people should be implemented. That can help in terms of them stopping, knowing when to stop, and yeah, that's what I can say. Thanks so much.
Thank you. I've got 3 minutes for questions. I've got 3 minutes for questions. So I'm not going to stop you unless you finish your 15 minutes. So, but I will only still be limited by question on that 3 minutes. Any question? Okay.
I have a question on the vaping. I understand that it is currently seen as a way that learners are supplementing their tobacco use by using vaping, and it has also been shown to be addictive. So I want to understand the rationale behind excluding studies that were addressing vaping.
Do you mind if I take questions and you just respond all of them? That will help you a bit. Because I saw there seem to be more.
All right, thank you for the presentation. My questions, they are just two in one. Number one is, what are the effects of this tobacco use in health system? You know, when a lot of youths, they have been using it for a while, you know, it's going to have health issue. Then what are the effects? Okay. Two is that, what about the economic manipulation of that usage of tobacco? Have you tried to work on how the usage of, or excessive usage of tobacco is affecting the economic power of people? So that's what I want to ask about.
You've got 1 minute. Someone needs to respond to questions.
Okay, I'll start by answering the vaping, right? when we look at the vaping, honestly, we don't know. Remember, sometimes they add tobacco, sometimes they don't add. So there are studies that cover that part, but we don't have much studies in South Africa. So these studies were only focusing on South Africa from year 2000 to 2023. So when we were searching the databases, we didn't come across such studies which will be talking about vaping. And our target group were adults and were adolescents and young adults. So among that group, we could not find such studies which will be added to our systematic review. So yes, but it's something that we will look at in the near future also, because it's really affecting our young people. Yes. Sorry, I don't know. Okay. Yeah, maybe the second part of your question, right? To say you're asking how it's affecting the health system. Sorry.
Health system. System.
Yeah.
The health system of South Africa. The second leg of the question is
Economy.
buoyancy of the people.
Yes. So obviously, when it comes to tobacco topics, we have to focus on one side, right? That there is economic side of tobacco and there is tobacco use side. So that side, I'm focusing on the use side without involving or without including the economic side of it. So yeah, that's what I can say. I've not looked at that.
Thank you very much. If some time, if there are some missing questions towards the end, I might allow some questions to come back again. I want to try to be fair for the time. You can see that I can give you a few seconds, but I want to try to be fair so that everyone get the equal chance to, to get an award for this. Yeah, I'm trying to be fair. The next presenter. Just get. I'm using an online one. Let me just use. The next presenter is Is it Guy? Guy, yeah. So towards the end, I will give a chance for any presented question that you might still be interested in to give, so that the judge can be fair. I need to keep the time equal for everyone. Thank you. When you want to start, just raise your hand and I will.
Afternoon, colleagues. I'm ready to start. Yes. My name is Siphoka Zigcayi. I am a student at the University of the Free State. I will be presenting part of my PhD research. So this particular work is discriminating Vachelia karoo from cohabiting vegetation using in situ high-power spectral data and simulated multispectral bands. So encroaching woody plants, they reduce grazing capacity, they alter the functioning of the ecosystem, they displace indigenous vegetation, and they degrade savanna and grassland ecosystems. One such encroaching woody plant is Vachelia karoo, which is a thorny woody plant that belongs to the Fabaceae family. So Vachelia karoo is a leguminous plant, which means that it fixes nitrogen and it provides forage for goats. So Vachelia karoo differs in plant structure from small bushes to tall trees. Its occurrence has been reported in many parts of the world and also in Southern Africa, and of which in South Africa it has been reported in various provinces and particularly in the Eastern Cape, where it has been reported to be increasing and is degrading the savanna and grassland ecosystems. So yes, like I've said, in Eastern Cape it has encroached both the savanna and the grassland ecosystems. So the spread of Vachelia karoo can be caused by wind or by animal droppings. Vachelia karoo has various impacts. It has socio-economic and environmental impacts, which negatively impact the rural people and the livelihood of farmers. So with such impacts requires that these encroaching woody plants, they be monitored and managed. So monitoring and managing woody plant encroaching species, they require accurate detection so that you can be able to map their spatial distribution. Previously, methods which have been used to map encroaching woody plants have relied on visual inspections, which involve the human beings, which means that they are prone to inaccuracies. Also, such methods, they are tedious, they are time-consuming, they are costly to implement, and they are not practical for large scale. Due to those limitations, therefore, remote sensing is a smart alternative because of its high accuracy and also high spatial resolution. It's also time efficient and it involves minimal costs. So the aim of this study is to discriminate Vachellia karoo from cohabiting vegetation using in situ hyperspectral data and simulated multispectral bands. The specific objectives of this study were to determine the best season in which this Vachellia karoo can be discriminated from cohabiting vegetation, and the other objective was to assess the performance of this ground-based hyperspectral data and also the performance of simulated multispectral bands. And also to determine the important bands when discriminating Vachellia karoo. So this study was carried out in the Eastern Cape, in the grassland side of the Eastern Cape, which is the Mhlonhlo local municipality, and in the savannah side of the Eastern Cape, which is the Raymond Mhlaba local municipality. So in the study area, we obtained a spectral reflectance of different vegetation. So in the study sites, the types of vegetation that were found was grass, and of course the species of interest, Vachellia karoo, and there were also mixed bushes, and there were also forbs, and there was also aloe. So we took spectral reflectance of each vegetation using an analytical spectral device. What you see here on my right side is just a spectral curve of each of the vegetation. So to discriminate Vachellia karoo, we applied a classification method using the random forest regression, the random forest classification method, specifically using the leave one out cross-validation method. So coming to the results, here, this is a table showing classification results of, I forgot to mention that the data was collected during late spring season in October and early summer season in December of 2022. So from this table, we were able to determine that Vachellia karoo can be differentiated from the other vegetation using the spectral data, as we can see there with the 100% accuracies. And we were also able to determine that there were not much differences between classifying either using spring data or the early summer season. However, we were also able to deduce that the best results we obtained during the early summer season. Also from this, we were reporting on classification using, that is your ground-based hyperspectral data using all the bands, and then from all the bands, we then selected top 10 bands, and then we also reporting on the simulated Sentinel-2 data. So all the types of data performed quite well. There's not much difference between the ground-based hyperspectral data and simulated Sentinel-2 data. So here, these results, they show important bands when classifying Vachelia Karoo from cohabiting vegetation. From these graphs, we can deduce that when we were using spring data, the most important band in classifying was found in the different regions of spectral data. So in this particular results, we're reporting on the bands in the shortwave infrared band, in the shortwave infrared region. Also, below here, I don't know if you can see my cursor, when we were using Sentinel-2 bands, we also observed that the important bands was the shortwave infrared band. And also, during the summer season, we observed that it was the red band that was most important in classifying this Vachelia Karoo. So coming into conclusion, this study sought to differentiate Vachelia Karoo from cohabiting vegetation using hyperspectral data and simulated multispectral bands. This study was able to obtain satisfactory results with high classification accuracies. This study also, the results of the study provides insights on the remote sensing tools that can be used in discriminating Vachelia Karoo from cohabiting vegetation. Coming to the recommendations, this study sort of contributes, in fact, towards the Sustainable Development Goals number 15, where it looks at management of invasive species. And from this study, what we have observed is that there's an existing gap between research that is conducted and the actual communities that are affected by things like bush encroachment. And there's also a need for awareness to be made to communities that are actually affected by this problem, because it's sort of like their everyday life. Sometimes people do not, maybe they do notice the occurrence of bush encroachment, but maybe may not be aware because it's sort of like a natural phenomenon. But if it is not managed and controlled, it can affect their lives. Thank you.
Right on time. So you can have your nice 5 minutes of questions. Can I ask some few questions up front and then you respond to all of them? Will it be better? Okay, questions? I won't ask questions because I don't want to be biased through. I ask a difficult question and people can't answer. I want questions from you guys. It's your conference. I'm just trying to help to facilitate.
Hello, I'm Paul, by the way. My first question is, what is the rationale behind you choosing early spring, your spring season and early summer? Why didn't you choose to collect your data during winter or autumn? And my second one is the SDGs. Which ones does your study align with?
Any other questions? Maybe a second one? At least we need to have 2 questions. You say we must be fair. I'm trying to say you guys must be fair. Also ask a second question.
Okay, thank you so much for the nice presentation. My question is that about the validation method that you used for the validation method that you used. Which one we used in random forest? And the other one, did you use the red edge component or you didn't calculate the indices?
Thank you. I'm going to bring it to you and then you can respond.
Thank you for your questions. I'll answer question number 2 first. You asked which of the SDG goals was my study aligned with. As I mentioned that my study is aligned with SDG goal number 15, which refers to life on land, but mine is specifically looking at the management of invasive species. And number 2, you asked why late spring and summer season, and why not winter? We did not consider the winter season because during the winter season, vegetation is green, is dry. So with remote sensing, it's a bit difficult to extract any information from dry vegetation. So we considered the late spring season because we initially wanted to collect data in beginning of spring, but when we visited the site, the vegetation was still dry. So that's why we landed on October and December. And the other question was which cross-validation method I used. I used the leave one out cross-validation method. And you asked if I considered the red edge band. I did not compute any vegetation indices. I used the single bands.
Still 2 minutes. So. I'll keep your time.
Just an advice, because it's more like what you are answering to this guy, because if you use those red edge indices, they might help you to quantify those parameters you were targeting, because a part of them, they are very accurate. And also the answer that you are giving now, saying because there was more like there's nothing on the ground. I was maybe expecting that there was a much cloud during winter. Then that interfere the quantification of Sentinel-2. But if you can just try to integrate the Sentinel-2 with Sentinel-1, you might get some very accurate results even during the winter and during the summer.
But I must say, I'd like to point it out that as much as you mentioned that had I used, I think you said vegetation indices, I might have obtained, better results. Is that what you said? But from our analysis, the red edge band did not come through though as one of the important bands. But thank you for your comment. I will consider that. Thank you.
Thank you very much. Okay. One of the candidates, I think they missed the flight yesterday, Boboyan. We will put them on tomorrow's session. So we will move to Emrapia. Will you read just show me my hand? Okay. Exactly. So far we've kept 15 minutes fairly square on point.
Okay, afternoon everyone. My name is Khapia Monde. I'm from University of Pretoria. I will take you some journey from one of our projects in Future Africa under Aurora SFN net in one of our campuses under Fanapan. And here we were trying to understand what is happening in Africa. Then we developed this concept, land restoration, biodiversity conservation in sustainable food system in Africa. We're trying to come up with more like something that we are owning. Because here in the morning they were talking about we must write our own book. So it was more like something like that, trying to have our own history book. So one of the problems that we identified during that time, it was a land degradation. And maybe everyone can notice the land degradation around his or her places. And this land degradation is not only affecting us as Africans, but the entire world. As you see that with that wide percentage, only 16% of the land in Africa which is stable. The rest is degraded. It's either severe or moderate or light. So that gives us more a serious problem that we need to face. Then we check the drivers of this land degradation. One of it is the increase in population around the world, even in our continent and our country, South Africa. And also the diet changes. We see that a lot of people try to eat healthy and try to get some sort of a very good food. And that thing is also comprising our food system and also our land. And also the expansion of urbanization, where we see that everyone wants to go to live in towns. Then we are reducing the productive land to our houses. And also the deforestation, where we see a lot of people cutting the trees and also leave the area bare. And also the unsustainable agricultural practices. We learn that these practices contribute a lot when it comes to the land degradation, because you'll find people are practicing with agriculture, but they are not following the good guidelines. And also the issue of climate change, which is there in our mouth every day. And this land degradation causes some threats within our system. The first one is soil organic carbon, which we are losing every day. Each and every minute you are losing the soil. And also the salination and also the solidification. You will see most of the soil now are more acidic compared previously. And also the issue of nutrient imbalance. So where we see that even if you planted something, it won't come out because there is no nutrient to feed that particular plant. And also the issue of erosion. And all of these cause some sort of ecosystem and landscape deterioration, and also cause a food insecurity. We know that our continent is regarded as one of the food insecure continents. Although we have land, we have everything, but we are regarded as one of the continents that is in poverty. This is one of the causes that cause those in food insecurity. The other one is also the migration. You will see people are moving from their countries and moving from their regions, come to where there is some sort of a hope to live. This is not wrong. Then we know that there are policies, and our continent is good when it comes to policies. But the implementation part is the one that is strictly. We see that from 1972, there was issues of land degradation were raised. And go to Rio 92, where there was a head of states. They were agree and the commitments, but still this land degradation continued. Here in Africa, we saw a Malambo declaration in 2014, where the head of state from Africa were meet together so that they can come up with the mitigation processes and some sort of strategies to mitigate the land restoration and biodiversity. But still, we are facing the same thing. Then it takes us to the one of the Irish thinker who said the sender cannot hold, and there's no one can hear the alarming bell. So meaning we are losing the hope towards the world. Then, but we should not turn despondent because the weather is bad, or when the sun shines, we become triumphalists. So we need to find a way to feed the world. As Mark was said, humans need food, water, so that they can think and persuade science. And also, the Russian revolutionists also highlighted that in order for us to think, to exist, there must be food, water, so that we can able to think. Then we must find out a sustainable practices that doesn't consume or try to kill anything. And as Luridin told us, that sustainable must be something that generates anything but does not destroy or harm what is available. And it gives us some sort of a thinker from India who said, if you stick to the description, you won't practice what is described. So meaning if you focus on describing the sustainability, you are not practicing it. You will miss the point. So therefore, we must find a way for sustainable practices than describing on words and papers. Then, one of the things that we need to do is to restore our land. Once we restore our land, we're going to achieve 3 SDG, which is provide zero hunger and a good health. But if we are missing that point, we are going to lose a lot of people. We will see people going to hunger, and we'll see a lot of people migrating. But if we are trying to reach that equilibrium, then we are going to have some sort of a good system with a good environment, and people will live in harmony. Then with this study, we try to check the contribution of land restoration and biodiversity conservation practices, enhancing sustainable food system in Africa. Here, we use 3 databases: Scopus, Web of Science, and Dimension. The 2 Scopus and Web of Science are normally publishing around those concepts. But with the Dimension, which is a new one, we see that we must fuse it so that we can see what is also being happening beyond Scopus and Web of Science. We got almost 65 papers. They were screened using English, and between 2010 and last year, which is 2023. And we remove 1 duplicate, meaning those databases are completely different. So we use a Prisma approach in order to screen our databases. Then we go to our results. One of the things that we notice is that South Africa was the leading country when it comes to production, followed by Ethiopia and Kenya. And if you check in North Africa, you will see there's a less that is being done about this land restoration and biodiversity conservation in food system. Then it also shows us these results that most people are working in a silo. As you see that only the collaboration is happening between Kenya and Mozambique and Burkina Faso. The rest are working in a silo. It'll give us some sort of a say by one of the France scholar who said, "I think, therefore I am." So everyone around this concept is working in a silo and is thinking he is the one. Then, and also we also identify that most of our work is being done outside Africa. As you see, one of the leading authors is from America. So meaning we must own what we are producing and we must own what we have. Then we have different concepts. One of the concepts that I will highlight is food security and climate change, which is a basic concept, meaning each and every time when the authors are publishing there, they are using food security and also under climate change conditions. Then also, even if you can check the connection of food security and climate change, it tells us each and everyone is trying to solve the issue of climate change and food security. But here now we see biodiversity conservation. It's among those keywords. Then for the trend of words, we see that climate change emerged around 2013. And also food security, meaning they are new and people are now trying to consider those keywords and they are trying to mitigate the food insecurity under climate change condition. Then it will take us to a conclusion now, saying the work under climate change and food security has been done in Africa, trying to address land restoration, biodiversity loss, and food insecurity. The study also highlights the trend and research themes, providing nations that are more leading. Then it also takes us to one of the speakers in the morning told us that education can be used as a tool to transform as the mind, but now the education must be used to transform the food system through what we know and we get what we don't have. Then we propose those policies because among the document that we need to produce in this project was a policy brief and also the synthesis brief. And we propose that whenever government is doing things, they must also include indigenous knowledge and also strengthen the collaboration among each other countries and also invest in research among this concept, and also promote agroforest and agroecology so that we can have the best food system in our continent, and also emphasize the existing policies about the land restoration and biodiversity, and also focus on practical initiatives so that we cannot talk something that is not being applicable. Then, by implementing these things, African countries can work together in solving our African countries, except going look for something that is outside. Therefore, also the collaborative will also help us in order to get the network outside Africa, so that we can bring up, so that we can also ensure that we are doing what we want for our people. Then that was the end of my presentation.
You have 3 minutes for questions. Quickly, I'll take 2 questions. Isn't it? So you can respond.
Yeah, can you please slide back? I want slide back, please. Just keep going back. Yeah, keep going back, please. Yeah, keep going back. When I reach there, I'll ask you to stop. Actually, okay, this one in particular. Those colors, what's the meaning of those colors? That's question number 1. Number 2 is that, research is fine. Writing journals to move up in the ladder of academics is nice. But what are you planning to do, or what have you been doing in reaching out to the communities, people who are actually affected by all those things, to sensitize them and talk to them about how to reclaim the land degradation? Thank you.
Just another question quickly so you can respond to. Minimum 2 questions.
Thank you for the nice, enthusiastic presentation. I'm just curious, you mentioned climate change, land degradation, because there are also climate change denialists who are saying that, the Earth has never had so many people like we have before. And part of the problem is that, the food resources are declining because humans are increasing. So I'm just curious, what's your take on that? Because you haven't mentioned anything about the overpopulation of the planet. Thanks.
1 minute and a half.
Oh, thank you so much for the question. The colors is just to demonstrate the distribution of published articles. If you see the dark red, which represent almost 28 number of publications. And what you were asking about the community, part of this project is more like a community engagement part. We were in Malawi last month, where we were trying to check what they are doing there. We also come from Botswana. We're also trying to check what they are doing there, so that we can sit down, then we strategize what we can help them, because these are the people that need the practice, not us as the researchers. But we are also doing that with the policymakers and the government officials, because and also extension officers and the researchers among that region, among each country, because we are also representing South Africa, and they also have some sort of a team there. So to answer you, it's more like it's a collaborative thing. And we are also working with the rural people and ensure that those land are being rehabilitated. And just to answer you, I mentioned that one of the causes of this degradation is the increase in the world population. I also put some sort of the numbers of the increase of world population, where we see that we need to feed that population as much as we also need to rehabilitate the land. That's why we talk about the systems. System is not only killing one thing or trying to improve one thing. When you improve this one must also stay positive.
Thank you. We are on time, 15 minutes to be transparent. I'm trying to keep the time. You will get the time for questions, but I want to make sure the judge is fair, on the time allocated. We're on time. We are moving to S. Is it Kaba?
First things first, I want to thank all the companies that made this project successful. And my project is on the yield response and nitrogen use efficiency of Mandela potatoes to cover cropping and different or selected nitrogen application rates. To give you like a little background of potatoes, they are very rich in starch, which makes them more staple in most regions like South Africa. They can be consumed in different ways. You can boil them, you can make fries, you can also okay. You can boil them, you can make fries, and you can also make a mash with them. Okay, they can be grown in a variety of climates and also soil types, which makes them more of I'm scared. Okay. They can be grown in a variety of climate and also soil types, which makes them more of the variable crops, and also farmers can be able to produce more and sell more. In that case, it's economical beneficial. Therefore, the rising global temperatures and also climate change has led to increased input cost. So in that way, farmers are now running at a loss because they have to improve production by increasing or by applying more fertilizers. So they just buy more fertilizers to improve production. Therefore, it is very important for farmers to increase production without buying more fertilizers or without with the available resources, and also those resources must not be a negative impact on the environment. In addition to that, nitrogen, in addition to potato production, nitrogen deficiency is also affects the overall plant production, because they affect the number of tubers per plant and also their size. So to overcome that, it is very important to apply fertilizers. So use fertilizers efficiently, like apply fertilizers in different growth stages of the plant, rather than applying the fertilizers at once. That is what we call nitrogen use efficiency. In addition to that, we can alternatively we can add we can use cover crops. Cover crops are very important in increasing nutrient in the soil, especially nitrogen, because we know that they are able to fix nitrogen. Okay, so a reduced fertilizer application due to leguminous or cover cropping system will then save production cost and contribute like a great deal to the sustainable One Health development goal. Therefore, and we also know that the cover crop and in-season management have been promoted as the key in-field practices for meeting water quality goals, but their impact on crop production remain purely quantified. Therefore, in this case or in this study, we then wanted to assess the impact of cover cropping and different nitrogen fertilizer application on the nitrogen use efficiency and also yield of Mandela potatoes in this case. This is our experimental design. It's a split plot design that has 3 fertilizer application, which is N1. N1 is 100%, N2 75%, N3 is 50%. And then we had to randomize cover crop and non-cover crop within blocks. And the overall field was 527 meters squared. Okay, these are our methods. Hey. Okay, we planted cowpea last year in November. So before cowpea planting, we took soil sample. Those samples determined the recommended rate for cowpea fertilizer application. And then we planted cowpea, and after planting cowpea, we then waited for 55 days. At 55 days, we then incorporated cowpea into the soil. We know that at 55 days, that was when the cowpea was at its flowering stage. And we know that the noodles are at peak to fix nitrogen. We incorporated cowpea for 2 weeks, then we took soil samples again. After taking soil samples, we then planted potatoes based on the recommended rates again. And then we had to take the parameters or collected data within that season, of which you'll see the next slide of on which parameters we took. Okay. And then we harvested potatoes, and then we processed data. Okay, these are the parameters that were measured for this particular objective, because we collected like a lot of data, but for this one, these are the ones that we collected. We collected the chlorophyll content, of which we used the chlorophyll spot meter. We collected the fresh tuber yield at harvest, and then we had it was in tons per hectare, and then the marketable yield in tons per hectare. And we also calculated the nitrogen use efficiency, which is YN. YN in this case is total fresh tuber yield, and over FN. FN in this case is total nitrogen applied. Then we analyzed that using our GenStat 23rd version. These are our results. The results that we got shown that these are our results for chlorophyll concentration. Okay, the result that we got shows that the plant started at a higher chlorophyll concentration, and it decreased over time. Of which, in this case, this is just the general plant growth, because you know that when the plant grows, it becomes green, and then it dies, and it becomes brownish, of which chlorophyll is normal. Okay, no significant difference was shown in all parameters. This was because we know that we incorporated cowpea. So when we incorporated cowpea, okay, we incorporated cowpea. So therefore, the cowpea might have improved the fertilizer in the fertilizer or nutrients in the soil, which then made other fertilizers like 75 and 50% also improve. And then this is the fresh tuber yield. It shows that there was a significant impact on the fresh tuber yield, and the highest yield was found at 75, at cover crop 75%, and the lowest was found at cover crop 50%. And then we found marketable yield, of which no significant differences were shown in all treatments, but the highest was found at cover crop 75%. This was because of the diseased tubers, the deformed tubers, and also those tubers that had shape, yes, this shape, yes. And then we then did that, the nitrogen use efficiency. Based on the yield, we saw that the highest yield was found at cover crop 75%, but it was not significant. Go back. It was not significant. The yield was not significant for non-cover crop 75. We then wanted to know if the nitrogen was efficient at cover crop 75 or non-cover crop 75 in this case. So we had to run the stats for that, and then we saw that the cover crop played a significant role in improving the nitrogen use efficiency of the moderate fertilizer application. With that being said, we then concluded this is just a preliminary study. We are going to run again the next trial. So, okay, the combination. So we then found that the combination of 75% fertilizer application, including cowpea cover cropping, had a significant greater yield of this much, 51.68 tons per hectare compared to other treatments. And then we also find tentatively, bear in mind that, okay, the study demonstrated that incorporating cover crop into the soil before planting Mandela will then reduce fertilizer application by 75%. This will help farmers to save fertilizer costs. Thank you.
You've got 3 minutes for questions. No, if you don't get question, it reduces marks for you for winning there. Question, 2 questions minimum. Everyone would have to get 2 questions at least.
Sorry, maybe what I want to ask, you might have presented when I stepped out. So I wanted to understand, in terms of your analysis, did you analyze the soil before the fertilizer? Okay. So, and also I wanted to understand, so when you check the nutrients, you check the nutrients of the soil before you applied fertilizer. Did you also analyze what could have been the contributing factor in terms of
after? Okay, you're saying before planting cover crop and after?
Yes.
Okay, it's improved. It's just that I didn't put the thing in. The results here, but it did improve the fertilizer in the soil, the nutrients, in this case, in the soil. Incorporating cover crop did improve. Just that I didn't put, just that I didn't see them come out, but it did improve. Why was it?
No, I don't have a question. I have a comment. I think this is a very good detailed study, and the fact that you're saying it's a preliminary study, it looks like you're doing amazing work. Thank you. So congratulations.
Thank you. You still have 1 minute. If there's burning question, within 30 seconds question. Okay. Thank you.
Thank you.
So guys, when I stand up, it just means that it's 9 minutes for 1 minute to your presentation time. But I don't stop you. I can only stop you at the end of 15 minutes. However, if you don't get a question, it means that the judge won't able to give you any questions, any score on the what? On the mark, on you responding the question. So you end up getting disadvantaged. That's why I stand up. But I won't stop you until your 15 minutes is done. It's just for me to say your time for presentation is ending. You are now chowing your questions time. And if you don't get questions, it affects you on the judge. Do you get what I'm saying? So don't be intimidated. I just want to make sure that everyone is fair. Okay. So we're going to go on the last speaker of the day. Then after that, I'll open it up for learning so that we are all got a fair chance. Thank you.
All right. My name is Timothy Oladiran Ajiboye, and I will be talking on investigating the performance of silver bismuth sulfide and graphene oxide incorporated into silver bismuth sulfide as tetracycline degrading photocatalyst in water. So I'm presenting on behalf of other co-authors that we work together on that. All right. Do you know that it is possible for you to wash your teeth 3 times in a day and still have colored teeth? Yeah, when we wonder, something must be happening. So one of the things that has been identified, apart from other things, is the presence of tetracycline in water. We wondered, why would tetracycline be in water, even after the treatment? Frequent detection of pharmaceuticals in water bodies is influenced by their high excretion rate. After you have taken the tetracycline, after the animals have taken the tetracycline, you discover that they will be released into the environment through the urine. And tetracycline will remain in the ecosystem even after it has passed through water treatment stage. You discover that even ordinary boiling water cannot even remove tetracycline. The implication of that is that if you are taking a cup of tea inside your house, you are taking some dose of tetracycline inside the tea. So, we think of the problem. How can we now solve this problem? What is the solution? One of the solutions is the use of light, sunlight. And sunlight cannot be used directly. There are materials that are needed to trap the sunlight so that they will be able to assist in degrading tetracycline in water. We call that term photocatalysis in science. Photo means light. Catalysis means catalyst that can help to trap light. So what we've done is to look at, okay, what is the research on tetracycline and other pharmaceutical removers, especially in Africa. There was a particular group, Mandela, in the year 2020, that did this research and look at, okay, what are the things, what are the drugs that are available in the water, even the treated water and surface water and so on. They took different countries. It was observed that 60% of the research that has been done on that is from South Africa. And analysis from the South Africa water show that all these are detected in those water. And part of that is the antibiotics. Among the antibiotics, the most common is the tetracycline. Now, that is why it is very, very important to look at how can we remove this tetracycline from water. Different ways people are using tetracycline. It has been discovered that millions of tons of tetracyclines are being produced worldwide in a year. Out of those millions that has been produced, just 200,000 tons are being used in a year. What happened to the remaining 800,000 tons and above? They are being discharged into the environment. And after being discharged into the environment, they will find their way into the water bodies. From these water bodies, we get our water, we pump water into our household. By the time you are taking that water, just like I said, it means you are taking some dose of tetracycline. I think you are enjoying the tetracycline. All right, we need to know what is photocatalysis. Photocatalysis, the principle is where you have material, every material, semiconductors, they have what we call the band gap. The band gap energy is you trap the energy from the sun, and by the time it traps the energy, there will be movement of electron from what we call the valence band, VB, to the conduction band. By the time it moves from there, there will be the electron that is moving will attack the oxygen to generate peroxide radical, and the hole that is left behind will attack the water to give the hydroxy radical. It is the hydroxy radical that actually attack the tetracycline that is in water and converts it to carbon dioxide and water, which is needed by the system. And that being said, we now think, okay, let's look at different materials. Different materials have been synthesized in the synthesis, to remove and trap the sunlight that will be needed for this tetracycline remover. What we've done in our lab here at Nelson Mandela University is to use silver nitrates with bismuth nitrate, and we use a solvent. We left that at 24 hours using DI water, deionized water in autoclave for 170 degrees centigrade. We monitor that, and after that, we get the sample that is formed. We are able to wash it and dry it. Then we take that for characterization to see what we have produced. Then apart from that, we look at graphene oxide. Graphene oxide, we produce that through a method called the UMA method. Because of time, I will not be explaining that one by one. So we use that method to produce the graphene oxide. Now, what we've done is to marry the two, graphene oxide and silver bismuth sulfide. The question is, why the marriage? Why not just using one alone? We have discovered that graphene oxide is the graphite compound. It is able to adsorb the tetracycline, to attract it from water. Why silver bismuth sulfide is a good photocatalyst that will be able to degrade. So we are looking at absorbing and degrading at the same time, simultaneously. So that's why we have been able to combine the two together. Then, from the result, little result from the characterization, we've carried out the XRD. XRD will enable us to know, have we been able to form that particular compound, and the result shows that we've been able to form the compound. We went further to look at the morphological characterization. We look at SEM, we call it surface morphology, to know what is. We discovered that the particles of silver bismuth sulfide is having circular shape, and we went further to look at the EDX. EDX were able to tell us the real elements that are in it, in silver bismuth sulfide. So we were able to discover that, no, we really formed the silver bismuth sulfide. After that confirmation, we did that for graphene oxide before we combine them. After the combination, we now look at, okay, let's look at Tau plots to see which one is having band gap energy that is closer to the feasible or in the feasible region. We discovered that when we use the silver bismuth sulfide alone, we were able to get 4.1 electron volts, which is far from the feasible. And by the time we combine it with graphene oxide, the band gap energy reduced. Why are we saying feasible light? We know the light that is common, if you look at electromagnetic spectrum, the larger portion is in the feasible region. And that is the region that we are looking at, that we will use for the experiments. So what we've done is to look at simulated sunlight. Because if we put it in the open sunlight, we'll not be able to monitor what is happening, what will be the intensity, and so on and so forth. So we try to fabricate a simulated reactor where we'll be able to monitor the lights that we are putting inside. So the tetracycline were now introduced. This is the point where we are introducing the tetracycline, introduced into the sample, and we're looking at the pattern of degradation after 1 hour. Now, what we find out after 1 hour was that when we look at the light alone is degrading, but it is very, very slow. And at the end of the day, we got something less than 30 after that 1 hour. It means light alone is not very, very effective for degrading tetracycline, though there is photolysis that is still happening. Then when we introduce graphene oxide, we discover that within that 1 hour, it increased a little. Because graphene oxide, as I told you, is like an absorbent, it can absorb. So that will be the reason why it has increased. But by introducing silver bismuth sulfide, silver bismuth sulfide is a semiconductor. It was able to degrade the tetracycline that is in water. But when we now marry it with graphene oxide, we are combining this with this. We discovered that there is a drastic and significant increase in the amount of tetracycline that was degraded within 1 hour. And from this particular research, we've been able to publish at least two papers from it, and one of them is having more than 5,000 citations, or read already, with over 5 or 10 citations. And I will need to acknowledge some people here. We need to acknowledge the next Mandela University, because they give us the platform to do the research. Professor Sabelo Langada is my host, and the chemistry department, which we use the platform, organizers of this Young Scientists Conference, Dr. Priscilla Mensah and Mrs. Wema Van Aden. No, we are acknowledging them because I'm a postdoc. So they were able to give us at least the platform to be able to do our research. These are some of the references that we use. Now, what we are suggesting is that if this research if we are serious with it, though we are partnering with the Municipal Bay so that we do real water analysis after the laboratory research. So, and that is another thing we are doing, is to look at what is the mechanism that is taking place inside the water. So we are currently doing scavenger experiments. Another thing we are still doing is to introduce algae into these composites, because algae itself, so some people said it can degrade. So we have read in the articles. So we want to now incorporate it into this particular material and see what will be the effects of introducing algae. A MSc student is currently working, her name is also here, on that particular project. Then the next thing is for the future reference, look at how this will now be produced in large quantity and how it will be applied in the commercial quantity, so that Timothy and people looking at Timothy will be free from drinking tetracycline in water. Thank you.
Still have your 4 minutes, okay. Or 3 minutes, sorry. I'll open for 2 questions. It's 12. It was still 11, still moving, so.
Okay, so what I would like to know is just if you can speak to why there is such an overproduction of tetracycline. And also, if you can speak to the probability of antibiotic resistance, since you are saying that we are drinking the water and by extension we are drinking the tetracycline in the water.
All right, thank you very much. Let me quickly answer that.
Let me just take 2 questions so that you're
okay.
done.
Sorry, can I just write them down?
Yeah, thank you, sir, for that fantastic work. My question is, let's look at it holistically.
Okay.
If you say you have a lot of tetracycline, because from this work now, I'm getting to understand the concept of tetracycline very well. Because all I know about tetracycline is what we use. Now you are now saying tetracycline is in water, then I'm surprised. Okay. Good. Now, we know our mothers, our forefathers, they have been drinking this water and they are very strong. Now, now that you are saying we have tetracycline inside the water and we are drinking it, have you tried to connect this water to any disease around us? Because if you are unable to connect it, then I won't see any reason why this. Yeah.
Okay, all right.
So I want to know if there's a particular disease that you have been able to link with this water containing tetracycline. That's one, please. The second thing is that have you considered the economical effects of trying to remove the tetracycline? And if you consider that, what about the locations, people living in the internet, how is it going to affect them economically too? Thank you.
All right, thank you.
Yeah, let him respond to these two questions, then I'm going to open the floor again for the last 15 minutes for everyone who presented.
Okay, sorry, I couldn't get the second question that you... The first one, you talk about why are we having a lot of tetracycline in water? Is that...
Why there is such a lot of tetracycline that is being produced in the first place? Because
okay.
You're saying that there's an overproduction of the tetracycline and then it's being disposed of.
All right, let me use this to answer that first one. Now, if you look at this, we have high demand of tetracycline in pharmaceutical industry we produced for the hospital, and human beings are consuming tetracycline. You know the population of human beings, we have around 7 billion. So you know the population that is taking tetracycline. And animals, different animals, no? In fact, some we introduce it into the fish ponds. Do you understand? So they are producing it in this large volume so that they will be able to meet, because the demand is very high. So due to that demand, a lot of it is being produced every year. Okay.
The second portion of the question was, is it not now introducing antibiotic resistance in humans specifically?
Okay, one of the things, the importance of this photocatalysis is that if you read very well, go and read, you discover that there is what we call inactivation of microbes. It's one of the photocatalysis, the application of photocatalysis. So, in fact, one of our research, we've been able to do that, where we use zinc oxide to inactivate bacteria, both gram-positive and gram-negative bacteria. So, apart from degrading tetracycline, any microbes, because from the explanation of photocatalysis that I explained, hydroxyl ion is very toxic to any microbes that is in water. So it's also doing that. We have done a research on simultaneous removal of microbes and organic pollutants. We have done a research on simultaneous removal of inorganic and organic pollutants as well. So it is also dealing with any microbes that are in water. Generally, even without thinking of all these emerging contaminants that are not being removed by the treatment method, because we classify them as emerging contaminants. They are emerging because they were not taking care of them before now, until they discovered that even after the treatment of water, they are using light. In the commercial place, they set light and think they have degraded all the pollutants. Okay. All right. Let me move. Sorry. Do you talk about the economic effect of removing tetracycline? Is that the question? Okay, yeah. When I opened, I talked about tetracycline coloration. You can browse it. You discover that. Apart from there are other diseases, which I may not be able to recall all of them now, because when I was preparing, I read about other diseases, but since that's not my focus, so I will not be able to be giving you all there are very many. And apart from that, it has been discovered that tetracycline water, people have been talking about sustainable agriculture. If the water contains tetracycline pass through farmland, microbes that are in the soil are being affected, and it will affect the yield of the crop. Do you understand? So that's one of the effects of tetracycline. And talking about the economic effect, every drugs are poison, quote and unquote, if they exceed the limit that is needed by the body. Do you understand? Every drugs are poison if they exceed the limit that is needed by the body. So by the time you are taking a particular, by the time you are taking it in water, you are not quantifying what you are taking. Some will take 5 cups of water per day, and they are even encouraging, drink more water. The more you drink water, the more you take poison. It is well with us.
Thank you. Thank you. So I've given him 2 extra minutes, so I'm open for 2 minutes for each. I may have 8 minutes left before we go for the, cocktail. So if there's burning question for any of the people that you still accept him because he already taken his 2 minutes on this 10 minutes extra. I'm saying accept him, I already given him his 2 minutes extra. Unless there's no other question for others. Yeah, but can I also make sure that other people get a chance so that there's fairness? I already given it. It went for 18 to 19 minutes, 18:56. So it was already 3. Unless there's no question, I will give. Yeah, only for the people who presented before. If there's a burning question that was still missing before. Unless there's none, we can go. We will give it. I just want to be fair.
Okay, thank you. My question, I want to ask the gentleman in front about the review that you did. I'm just curious, I heard you mention something about the limitation when you were searching for the papers, but I'm still curious that did you manage to make a scientific conclusion out of 12 publications? Because I think the minimum should be 20.
Okay. I'm not sure about the minimum of 20, but the paper is published as we speak.
Okay.
So it was accepted as it is. So though it was very difficult to get all those publications because we were very specific about it. So we're looking at the people who have included the prevalences in their papers. So we're looking at the tobacco use as in general, but we're very specific about it. That's why I put some of the products like I didn't include some of the product because some of the product won't know if they've got tobacco in them or not. So about the minimum of 20, the journal didn't disagree with the paper, so they published it. Okay. So yeah, the paper is published.
All right, thank you.
Thank you.
I'm sorry, I just wanted to comment on the minimum. There is really not a minimum, and also there is not a maximum. You can actually have a systematic review done, and then you actually have 0 studies included.
Thank you. Any other question? You can ask any other question. Still have a...
Yeah, I would like to ask a question from what that beautiful lady presented the other time. But I want to ask her if...
I think I see all the beautiful ladies.
No, I want to ask her if she want to answer me.
Eh?
No, this lady...
You can ask her questions.
No, I need to ask her.
Remember, some of the things here we are learning, guys. We need to learn from each other.
If she agrees to answer, I will continue.
Okay.
If she agrees... she actually presented work on the food I like most.
It's also a time for learning, guys.
Okay.
It's not about questions now. You're not going to be affected. This is for more learning. The part is done for the award. Okay. It's collating. This is for learning so that we learn from your work. Remember, the whole point here is to come to learn from each other. So I'm giving you space. So if you have a question you want to learn from your work, just give him a response.
I'm still learning myself.
Unless you want to talk through in the cocktail time. Okay, I just want to give you a bit of what's going to happen. So what normally happens is that we've got a few numbers of postdocs. It's for the first time we have added postdoc in our conferences. These conferences normally focuses on masters and doctoral students, but obviously postdoc, they are in our radar because postdocs are the only ones that qualifies to become SciAS members because you need to have a doctorate to become a SciAS member. But the conference in the past used to be the conference to give capacity for the masters and doctoral students so that we support them nationally. That's why we have 29 universities that are represented here. So because this year we've added postdoc will be evaluated as postdoc on their own. So we will look at all the sessions of postdoc separately. The hours for postdoc will be separate from this so that there's fairness because postdoc are more experienced. They have enough. So when we do the scoring, if it's postdoc, it will be reviewed in alignment with other postdocs so that there's fairness. That's why you could see even on the timing, I was trying to make sure that everybody, at least if you bypass my 15 minutes, it's by a few seconds so that we are all fair. You got 2 questions or at minimum everyone so that he can able to do an evaluation on the part of how you answered the question. Because the east part, if you don't get a question, then you get disadvantage. So the only one way I could stop you from presenting, if you present the whole 15 minutes, I would allow you. But obviously, you are going to be affected on the questions, so you're going to get 0. Do you get what I'm saying? So that was the thing when I stand up, I was trying to say your time for presentation is finishing. You are about to take in your questions. And if you don't get question, you're going to get 0 on the section of questions. but I won't stop you from presenting. Okay, so I just need to give clarity for. Are you calling for cocktail? Don't worry. You can also that's why I make sure it's fair. Even the last person of question is not part of for the review. That's why you see I try 2 minutes, two questions each, to make sure that there is fairness. Now remember, the conference is not for winning. The conference is here to learn from each other. So I wouldn't stop the learning. That's why the reason we had enough time, because one person didn't present. You see, so you can still go now to the cocktails, continue the conversation. If you want to talk to the beautiful lady about potatoes, you can talk to the beautiful lady while holding cocktail. You got what I'm saying? Okay.
I'm just worried. He's saying that if we continue drinking water, we are. So I'm really worried about myself, because as a health professional, we are told to drink 2 more cups. So if you are saying the more we are drinking water, the more we're going to die or something like that, I'm really worried.
Yeah, the more you are drinking water that has been polluted, I'm not saying all the water has been polluted. The more you are drinking water that has been polluted with tetracycline, the more it is dangerous to our health.
So I must put it in the sun.
No, you can't. Until you do there are things they are doing in the municipality and some other places where they are treating water. So until they have done the treatment, it was recently that they are discovering that there are other emerging contaminants which they have not been thinking about before. Example is microplastic. I don't know if you have heard about that. So they were not thinking about microplastic before. So later they discovered that, no, we have to look at this in detail. So the more they are having more sophisticated equipment, the more they dictate some of these things. So by the time you are buying the so-called pure water, so pure water may not be pure.
Made a clarity, and I think probably I was fair, am I right? Because I'm from the South African Young Academy of Science, I needed to be fair. That's why I didn't want to ask questions. I needed to make this conference your conference so that you ask questions yourself, and to make sure that every God get a fair chance. So that, when awards comes tomorrow, don't say that guy was favoring that guy, is what I'm saying. That's why they got an award. So that's why we are trying to be as fair as we can as the South African Young Academy, because this is a flagship conference that gives capacity for all of you guys. So thank you very much for the presenters, and thank you for the support system on that side, and for our cameramen. We can clap hands for them. They did a very, 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 very good job. Thank you very much. For those who are presenting tomorrow, try to make sure that you give your presentation, early in the morning, whatever, so that your thing is already recorded, so that you can start on time. I'm going to chair some sessions tomorrow again. I don't know which ones, but but I try to be fair. Thanks. Thank you. Can we have some cocktail and discuss about potatoes?